Then we have done some biological characterization where cell culture studies are done, where because our interest is to take this forward for bioprinting of cornea, where cells will also be there encapsulated within the hydrogel. So very first thing we try to understand whether the cells survive within this bioink or not. If you clearly see here, this is the live did staining here, where at day 1 we see cells are very much viable, and also upon with days the number is also increasing. K67 staining typically provide you the idea, provides you the idea that the number of proliferating cells within the matrix. So these are the way the cells, this red color thing, this thing are those are the proliferating cells. As you can see, the cells are also proliferating well till 7 days, then the number is getting reduced. So here, if you see here, this is the NPTEL-NOC data where we have compared this our cornea matrix hydrogel along with the standard material like collagen, and also in 2D. If you clearly see that the DCM is till 7 days, their proliferation is similar. At 14 days, the cell number is high in collagen but is less in DCM, and also in 21 days again the cell number is getting lowered in all the cases. So typically, because keratocytes, when you culture them outside, they lose their phenotype, can become myofibrous like cells, and they can grow very fast. And also, but upon time, the keratocyte cells upon time, their numbers generally reduce, because in tissue culture plate outside the native tissue environment. So for human cornea tissue, digital cornea matrix hydrogel, also similar thing we have observed. In 14 days the cell number is getting increased, and also cells and a cell number, they are highly viable. But K67 at the initial days they were proliferating higher, but the numbers is the proliferating cells number is getting reduced with time, and that is typically a characteristics of the keratocyte cells upon in vitro culture. As see, these are Ki60% positive cells, as that cell number is very high. The polytonic cell number is very high at the beginning, but that is upon time that is getting reduced. The other thing we have understood, we try to understand that whether this hydrogel can support the cell migration within the gel or not. Suppose if we put cells within the gel, whether the cells would be able to migrate within the gel or not. So one way to understand is the wound healing assay. Typically, in wound healing assay, what we do, we first we culture the cells in a plate, tissue culture plate, and then grow them to full extent. And then when the cells are fully grown and confluent, there we create a wound with a pipette. We can create a wound, right, like this, so that the cell numbers here, the cells are gone, and then two fronts of cells are possible here, are present here. And now these two front, whether they can able to bridge the gap and they can meet that thing upon time, that is the wound healing assay. Now here, if you see, without, when we put nothing is there, scratch is maintained, scratch is done, or scratch is made, then you see the cells, both the sides cells can migrate, and they can bridge the gap in 24 in 30 hours. The no presence, the scratch is even not visible, so we cannot find where the scratch is. So that means the scratch, these cells are highly active, and they can migrate and bridge the gap. But now we will try to reproduce the same thing with our hydrogel. So what we did, first we made a scratch, then put our hydrogel in between the gap, and then we try to see that whether the cells are down, it is the cells, whether the cells are viable or not, and also cells can migrate through this gap or not. What we found that when the gel is also there, the cells could easily migrate and they could bridge the gap. But in this case, we cannot understand whether the cells are uh, passing through the hydrogel, over the hydrogel, or below the hydrogel. So we did a confocal microscopy by which we can understand whether the cells are present within the hydrogel or not. Now in this case, you can see a depth dependent color is being given to the cells, and the cells passing at different heights is given a different color, different pseudo colors. So based upon that, from this thing, when you quantify, we found that almost 200 to 250 micron is the space where most of the cells are passing through this space, this height. Almost 80%, close to 80% cells are passing through. So this tells us that this hydrogel is highly functional, and it also induce migration of the cells towards this. It has typically it should have some chemoattractant present within this, so that's why the cells are viable, and they can also migrate within the hydrogel. Now the next challenging thing to work with human keratinocyte is that these cells often lose their phenotype and this thing, and they become myofibrous like cells, and they go very rapidly. But these are not typical keratinocytic cells. So they present a scar. They form scar tissue, and then they become contractile. They produce a lot of collagens, and then the tissue become scar scar tissue, and this is, but that is not functional. And in cornea, if there's a scar tissue form, so that can lead to vision loss. So that's why our interest is to whether, suppose if we culture these cells on a hydrogel, whether these cells can maintain their morphology or not, or whether the cells are becoming myofibrous or not. So what we did here, we coat the tissue culture standard tissue culture plate with our this digital cornea matrix hydrogel, and then we then you tried culturing the human keratocytes on top of this thing. Now suppose this in first panel, this is their tissue culture plate without any coating, and in this case tissue culture with coating. You can clearly see when the cells are cultured on this thing. Here typically they are like keratocytic morphology is present, but in this case over time cells are becoming elongated, and then typically they are becoming myofibrous like appearance. But in this case the cells typically they morphate like this kind of elongated morphology. Fibroblastic morphology can be clearly seen here upon incubation for 40 days 14 days. Also we have tried to characterize the same thing with by doing some gene expression analysis, where we have analyzed three different genes like collagen 1, alpha SMA. Alpha SMA is alpha smooth muscle actin, and also keratin sulfate. So these three genes we have analyzed, and we have compared with the collagen, that is the standard material for different corneal applications. So our DCM hydrogel, it can both the hydrogel like collagen and DCM. Collagen 1 expression is more or less similar, but alpha SMA expression is much higher in collagen gel than the DCM hydrogel, and also the keratin sulfate expression is much higher in the much higher in cornea matrix DCM hydrogel than the collagen, only collagen gel. So if you see alpha SMA and keratin sulfate, these are the two. Alpha SMA is a typical hallmark for myofibrous cells. When they become myofibrous, they express a lot of alpha SMA. So that means probably in case of collagen gel, the cells become more myofibroblastic, and also keratocytes they express more keratin sulfate than alpha SMA. So that's why we found that this DCM hydrogel can definitely DCM hydrogel can able to maintain or preserve the morphology of the corneal keratocyte morphology and the genotype. And again, we have done the immunocytochemistry study where we clearly seen that in the collagen DCM collagen matrix, DCM hydrogel, the cells are encapsulated in the DCM hydrogel. They express or they produce more keratin sulfate protein than collagen 1 and alpha SMA. That means these keratocytes, they can maintain their morphology and also genotype, and then they can express higher amount of keratin sulfate, or they can express. So that means these cells can maintain their morphology, and they can become more like they become more, or they can maintain, and so that they can function like even keratocytes, and so that is a good thing, good finding from the study. So that can be so taken care of by this thing. Then the other thing is also we tried to understand whether we measured the construct diameter. So construct diameter can be an indication. Suppose when these human keratocytes, they become myofibroblastic like cells, they try to contract the matrix, and because of that the diameter will be reduced because of this contractile force exerted by this myofibrous cells. So we have encapsulated this corneal keratocytes in our DCM hydrogel and also in the collagen hydrogel, and in both these cases what we found that in case of collagen hydrogel the construct diameter is reduced significantly, but in case of DCM the construct diameter does not reduce significantly. That means the in case of DCM, definitely typically the cells, probably the corneal keratocytes, they maintain their morphology and also the state. But in case of collagen hydrogel, they become myofibrous. That's why they become contractile and they try to reduce the dark matrix because of that. Also, we have tried two different concentration of FBS because it was found that if we use higher percentage of FBS, fetal bovine serum, then it induces this myofibrous transformation, transdifferentiation. That's why you have tried both two different percentage, and here you can clearly see that in case of this thing, when the less percentage of previous users, the construct diameter reduction is more. But in our case, like in DCM, both the cases, the construct diameter reduction is not that significant. That that means this here the cells can maintain their morphology and thing. Next, we have done some in vivo studies. So where we put this hydrogel in the uh, rabbit surgically depletion wound model. So because we try to understand whether this our hydrogel can induce tissue regeneration and also can prevent the scar tissue formation. Because what happens in cornea, whenever there is an injury, that injury can lead to scar tissue formation. Because in the injured cornea, here the cells, the keratocyte cells and that injured space, they become myofibrous and they put a lot of collagen, and that due to the uh, unordered arrangement of that collagen fibrils in that wound area, that area become scarred, and because of that the that cornea become miss the cornea become opaque, and people they lose their vision. So because of that thing is, so because of that, our interest is to use that our DCM hydrogel as a prophylactic and therapeutic strategy for blinding cornea scarring. What we have done here is that this is the overview of the study, where scarring leads to keratocytes, and the keratocytes, whenever the scarring happens, because the keratocytes they become myofibrous, and when they become myofibrous, they produce a lot of ECM, and those ECM actually they just dump the ECM in the, suppose there is here, this is a cornea, typical corneal scar where the whitish tissue can be clearly seen here. So because of this, the whitish tissue here, what happens, this myofibrous they produce a lot of ECM, and this ECM is here is a opposite arrangement of this collagen and other ECM molecules. Because of that thing, the it become opaque, and because of the fact this, the corneal scarring leads to blindness. So the corneal scarring leads to blindness due to this thing, and the cause can be many, including injury, infection, post surgical intervention, etc., etc. So that can be big due to that corneal scarring can happen. If you see the current treatment strategy, only cadaveric corneal grafting or full thickness transplantation is the only option for this kind of thing, where the part of that scar tissue is taken out, and and a, a corneal graft that is taken from somebody like this, a donated corneal graft, that is put here for to gain the transparency and to gain the vision. But there is no data till now, there is no study where some hydrogel or some drug is used as a prophylactic approach where it can prevent the scar tissue formation upon injury. That there is no such data available. And also there is no other hydrogel, other injectable or other thing, other form that is being also used for this kind of to prevent scar tissue formation. So in our study we have tried to do that thing. What we have done actually, we have created a wound here like something like this, and then we have created a wound, and then we have, by taking out the, suppose taking out the epithelium and a part of the stroma, something like this, and then we put our hydrogel in that wound area. Now suppose then we close that thing with a contact bandage lens, and then you try to see whether this hydrogel now can able to integrate within this microenvironment, within this environment, and also can lead to constructive tissue regeneration or not. So what we done, this is this before incision, you can see after injury where this thing, and then the hydrogel is placed here, and then you can clearly see his after hydrogel placing, a bandage contact lens is applied there, and then then it is kept under observation for 90 days. And here We have also to characterize this thing by various ways. One such way is the fluorescence staining, by which what we found that when this thing, like suppose at pre-operation, fluorescence staining is one such stain where if the endothelium is intact, that intact endothelium will not get this stain. But if the endothelium is lost, then the tissue will get the stain. So here, if you see clearly, see here at the pre-op where the endothelium is intact, here the no staining can be observed. But at J7, when the endothelium is gone, and then what we done, because due to the surgical depletion, the endothelium is gone. Then here, this group clearly they are taking this stain. But in case of this, either it can be human or bovine dislodged corneal matrix hydrogel. In both the cases, the tissue did not take any stain. So that means here the epithelialization or epithelial tissue certainly formed. Otherwise, this could have been taken, also taken the fluorescence staining. And over time, if you see at 90 days, almost of the tissue they got no epithelium. So epithelium is mostly whacked, and then the tissue got the epithelium tissue base. So no fluorescence staining can be observed. Then also we have done is these are digital photographs taken from camera of the rabbit cornea eye. You can clearly see here at pre-op the tissue is highly transparent. So there is no whitish tissue formed here. But at 7 days, if you clearly see here, the tissue is like here, the transparent whitish tissue appearing. So that is the scar tissue, and scar tissues are. And in case of bovine and human disulloid corneal matrix hydrogel, we can see that scar is slowly is minimizing, and then at 90 days we could not see any scar tissue formation, scar tissue in the our cornea, in the on the injured cornea. But in case of control where no hydrogel or no treatment was given, it the scar tissue is clearly visible. A white scar tissue upon 90 days. Even in we have compared with the collagen group where the after the injury, the collagen hydrogel was put there. Also, we can see clearly scar tissue was formed. So that's the typical. So that this two hydrogel, the bovine and the human distributed corneal matrix hydrogel, has something that is preventing the scar tissue formation. So definitely it can be used as a prophylactic material for prevention of scar tissue. And we have also done OCT analysis. OCT is optical coherence tomography, by which we can clearly see the tissue cross section. And you can see here if the tissue is opaque, that time a whitish spot can be whitish, this kind of things can be clearly visible. Suppose in case of collagen, if the tissue is also highly opaque, because of this thing there you see the white spot is very white. But in case of HDCM and BDCM, you see that white spot, it is though at the beginning there is a white spot was there, but at 90 days it is almost minimized. So that means transparency is gained. And in control, you see that one thing is the thickness is getting reduced, and the other thing is also the tissue, the white spot is very visible. So a scar tissue is formed. Also that check the thickness in both the, like in collagen and also in control, the thickness is also much lower than to the other HDCM and BDCM, and it is very similar to the native tissue structure. So these are the pachymetry analysis where the thickness of the cornea can be measured. And if you see here, scale is given here. Typically the cornea has a thickness of around 400 to 500 micron. If you see here at 90 days, if you just see that in most of the cases, in collagen or in control, the thickness is much lesser than that. But in case of HDCM or BDCM, the thickness, though at the beginning the thickness, because we put hydrogel, so the thickness was elevated, but after 90 days the thickness is brought back to almost like in around 420 to 460 micron. In that range the thickness is present. So that's a very good indicator for corneal tissue regeneration here. Then also we have done analyze the we have done histological analysis of the same cornea tissue here, and so that we can understand the micro architecture of the tissue. And you can see here, this is the native cornea where typical epithelium, endothelium, and stroma, and all the keratinocytes clearly visible in the stroma. So if you see in other tissue, like in other groups, like in collagen also, a thicker epithelium and an unordered arrangement of collagen fiber is present in the stroma. In case of HDCM, BDCM also, a typical arrangement of collagen fibrils is present, and also the epithelium is also more close to the native tissue. So that means this here in this case the tissue structure, the architecture is also very similar to the normal cornea. In case of scar tissue, you see the collagen fibers are arranged in a different way, in a different order, and also there is also the structure of the tissue. This tissue is not that close to the cornea. It's very different. And also we have done pus staining. Pus is a staining gives an idea about the presence of mucopolysaccharide within this thing, within our cornea in the thing. And as pus is the structure is, so that much thing, that much ECM component is present here. In case of normal cornea, you would see here this more pinkish than the scar cornea and the collagen group, and but the HDCM, BDCM is very similar to the normal cornea. So from histological analysis, our understanding is that this hydrogel, typically this hydrogel has, first thing is it prevent the scar tissue formation, second the transparency is gained, and third is the architecture of the collagen and other tissue, or the architecture, microarchitecture of the tissue is very close to the native normal cornea tissue. That means this hydrogel typically it has some bioactive component that helps in this constructive tissue remodeling and also the corneal tissue regeneration. The next, then after analyzing the studying the cornea in detail, after studying the visualized cornea matrix hydrogel in detail, also we have developed the Bioink out of it, where we have, as first thing we have done the physical characterization, rheological characterization, then the next thing we have done the also component analysis we have done, and then we have done the biological characterization, where we have done understand the cell viability, cell morphology, cytocompatibility, all these things we have done. Also the cell migration analysis also we have done, and the other thing is also we have done genomic and proteomic analysis to understand whether this Bioink whether this visualized cornea matrix hydrogel or Bioink supports constructive tissue regeneration, corneal tissue regeneration. In our in vivo studies, we found the same thing. Then we have adapted a bioprinting approach, where here we have used a mold based approach, where first the support mold. These support molds are like, these are like a convex shaped, dome shaped support molds were prepared as per the design, as per the corneal structure. Like these support molds are also almost like 12 mm in diameter, 11 point this mm diameter, and so these are stripped centrally in the peripheral, and then this thing. So we have used this concept, mold based concept, for generating this 3D bioprinted cornea. And these molds were printed by stereolithography technique, by which we can use some resins, and by using the resin with the laser light we can generate these structures. We can print 3D structures. So in this case the CAD model, these are the some details about the skit of the CAD models and the printed mold. This is the structure of the printed mold. So this is typically a dome shaped printed mold we have obtained, and then from there we have used this thing. Also another thing we have done, we have optimized the process parameters for this Bioink, as it is, as you know that the process parameters of the Bioink is very important, because every Bioink they have certain window where we need to work on, so that the pressure, because it extrusion bioprinted, the process parameters are the like the extrusion pressure, the pressure, pneumatic pressure, how much pressure we are applying on the Bioink, the print speed, extrusion rate, all these are very important parameters. Here we have optimized those parameters for this Bioink. Like if you see at 0.1 bar the material is underflowing, so that means typically this underflowing, so that pressure is not good. But at 0.2 bar the material is flowing through that thing, and we can clearly see a strand forming here. So that typically, so that this tells you that this probably this pressure is good for extrusion, and when the pressure is increased, you see the more material is coming out, so that is also not good. So from this we understand that, or we found that 0.2 bar this pressure is optimum for extrusion, Now, so that what is the print speed that is required for printing this kind of lattice structure? Now, suppose when we print with 3 mm per second, the lattice structure at the same pressure, this pressure, and 3 mm per second print speed. You see, the lattice structure is printed, but it is getting widened. The lines are getting widened, so the structure is not that great. But when in this case 7 mm, the structure resolution is good and the lattice structure comes out nicely. But at 10 mm, this speed is probably the higher side, because that's why the lines are discontinuous and we could not get a nice structure like this. So 7 mm probably is the optimum speed for printing with this pressure. Then also another thing we have done, this lines, as we have printed the lines like in the corner, when the lines are like getting the printed is going and it is taking turn and coming back. So that time here there is always there is a chance of widening up the line at that here. So we have to found that this range between 6 to 8 mm per second, that range where the lines are getting perfectly printed, but over that lines are not getting perfectly printed. So this is the window. And again, the circularity of the pores, like suppose when you print the structure at this speed, the prints are there, the pores, the lattice pores, typically this would have been rectangular, but in this case the pores are becoming circular. So that happens. So when the pores are becoming circular, so this way that their PR is less than 1, but at PR1 the surface says more or less it is rectangular, and it is when it is deformed from there it can be more than this thing. So we found that in between 7 to 8 mm speed, also the pores structure can be maintained, very well maintained. Then we have optimized all these process parameters, and we use this optimized parameter to print a structure like this, a lattice structure like this, to understand how does it affect the whole printing process. Then again, coming back to the bioprinting of cornea, a dome-shaped structure as we have mold we have created. Then we created a STL file, the CAD model of for bioprinting. Here you can see it clearly a dome-shaped STL file is made, and then that slicing another thing, or with here how this nozzle will move. So basically, in our case, the nozzle move from the outer way to inner way, outside to the inside of this things, or from the low top to the from the bottom to the top, or from the top, sorry, from the top to the bottom, thus printed will move. So this is the print before printing the mold. It looks like similar to this. Then we printed the structure within this, and after we printed, you will see clearly a hydrogel structure that is printed on top of this is visible here. And these are the print parameters that we have used, and this is the cell concentration. So we have used only 1 million density, and we have used human corneal keratocytes isolated from the cadaveric human corneas, present human cornea, and the speed is like this, and pressure and other temperature, other process parameters are given here. Now this is the printed bioprinted cornea, and you see this before and after. You can clearly see the bioprinted cornea is clearly visible here, right? And this is the cornea. And the one important finding from this study is this: we can produce corneas with where the this central part is less thin, less thicker than the peripheral part. Peripheral parts are more thicker, and also because of that, the you see here middle part, the cornea is more transparent here than the thing. So it is very good for handleability also, because uh, if the peripheral parts are of the outside part, that those are thicker, and those you can maintain the shape of the cornea, and also it can be very well for handling. So if you clearly see, this is the printed structure, this is the vibrant cornea. Also you have compared with the human cornea. This human cornea, as I mentioned, the diameter can be anywhere between 10 to 12 micron, but in this case, in our case, we have printed a almost 9 mm diameter bioprinted cornea here. Diameter is almost 30.56 to this, and after 14 days of incubation, the diameter is reduced to 12.16, so almost 12 mm of diameter cornea you have got. Height, sagittal height is almost 3.665 millimeter, so that must tip the central part of the cornea in our case. Then we have done some biological characterization, like whether the cells are viable within the bioprinted construct or not. As this is the very first study you should do after bioprinting, whether the cells are viable within this cornea or not. So that's the very first indication that will tell you whether the hydrogel is biocompatible or not, or the bioprinting process exerts any cytotoxicity or not. So you see this is not bioprinted when it is just plated without the bioprinting. And in this case, when the cells actually cells are cells that are encapsulated in the hydrogen, they are bioprinted. The cells are very much viable. In both this case, there is not much different. There is not much different. Okay, you can clearly see here the viability is almost 100% cell viability is maintained in in all the cases. Right? And also then we found that bioprinted cornea, this is the bioprinted cornea, this is non-bioprinted cornea, that swelling, diswelling that I was talking about earlier, that the only hydrogel when you put it into the buffer, they or media, they diswell. But in case of bioprinted cornea, diswelling is lesser than the non-bioprinted one. In terms of absorbance also, like with the cell, this is MTTSA, so where the cell number, if you see from the 1 day to 7 day, the cell number increased, and then up to 14 days, so the growth has reduced, but the cell number is keep on increasing. Bioprinted, non-bioprinted, both the cases there is less difference, but bioprinted cornea actually supporting more cell growth, probably alignment of the cells or something related to that. Then also we have done some histological analysis and also immunocytochemistry analysis, where we found that in the bioprinted cornea also the keratocytes, they can maintain their morphology and genotype and phenotype, because they produce a lot more keratin sulfate, less collagen and alpha SMA. So that's a very good indication that the cells here, they are viable, and they are also functional. They are producing the thing that is required for the keratocyte. Typically keratocyte specific things are getting expressed here. So those things are that is very important thing. And also that is the histological analysis of this thing, corneal analysis. Here see the collagen fibril arrangement is not up to that how it is present in the native tissue, but anyway they are maintaining particular order, and probably with time this can be further remodeled, and this can be kind of further remodeled with this thing. So this is an important finding from this study, that the cells are viable, cells are functional, and they can maintain their keratocyte specific functions. Also, the next thing we have done, also we have done the mechanical characterization of this cornea. We have done a compression analysis to understand whether the structure can prevent collapsing, and also if the structure can be disrupted upon force, what is the how much force is required. If you can clearly see this, we cannot compare the bioprinted cornea with the native cornea. Native cornea has very high stiffness, like it has a good amount of stiffness, so that it prevents collapsing and disintegration of the structure. But bioprinted cornea is like almost almost 5 times less than the native cornea. But please understand, this is just the cell-laden hydrogel. At this case, they have not matured. They have not matured, and there's also some kind of cross-linking and other things. Those are not yet fully regained. So that's why this tissue is not mechanically not comparable to the bioprinted cornea. But important thing is, if the tissue can be handled well, and if that tissue can be put into the if that tissue can be also put into the on the surgically depression site, they mishandled and transplant. If the tissue can be transplanted, and then at the initial stage, if we can control the attachment of this bioprinted cornea with the native tissue, and also if it can be successfully integrated with this, that's a very good thing. So because of that thing, this is so that these things are very these things further we are doing further studies from this study. Now we are also doing some in vivo studies, all these things, where we have successfully implanted our bioprinted cornea on the surgical on the rabbit wound, where we have created a 8 mm defect. That's a big defect actually created by surgical depletion, and then we have put our bioprinted cornea there, and we have seen the very early sign. We have seen that we could successfully implant the bioprinted cornea on the wound bed, where the bioprinted cornea actually can very well attach to the wound bed. And then we have put a contact bandage lens. Now we are doing further studies to understand whether this bioprinted cornea can be integrated with the existing stroma, and how does this remodeling happen, whether the then this thing, whether the bioprinted stroma at this stage, the bioprint after bioprinting we get up to a 70 to 80% of transparency we can achieve. But we hope that upon after in vivo implantation or after remodeling and regeneration, probably the rabbits will gain their vision, or that can transparent, the tissue will be transparent, the bioprinted cornea become transparent, and it could help to restore the vision in the patient. So these studies are going on. Also, simultaneously, we are excited about the other one, that the cornea hydrogel. There also we are looking for clinical translation of this hydrogel, because this hydrogel has got, as I mentioned, this hydrogel goes to very important properties. It prevents transdiffusion of corneal keratocytes, so it maintains the corneal keratocyte. So it prevents scar tissue formation, and also it can strengthen the cornea by mechanical reinforcing the thing. So because there are two major corneal diseases like scar and keratoconus, so we are targeting these two diseases for to treat with our corneal matrix hydrogel. So these are exciting development in this particular area from our lab, and we hope we could be successful in generating this bioprinted cornea, and that can be useful for to save vision for many patients. Because if you see the artificial corneal graft is in high is a high demand, and there is a huge requirement, and if we can successfully bridge that gap, that will be wonderful, and that would definitely that could then we can save many patients, and we can actually give visions to many patients. That is very much required. So in conclusion, DCM hydrogel has advantage over other existing materials, including easy availability, and we have developed a simple formulation procedure, and this hydrogel is highly biocompatible in nature, as we have seen from the other studies like the cell cell proliferation, cell viability, cell migration, and other functional analysis. And also other important thing is the hydrogel is that it prevents transdiffusion of corneal stromal keratocytes, and also so that uh, it prevents the microscopic differentiation of corneal keratocyte. So it can be used as a potential. It can be used as a material for blinding scar tissue formation. Also, it can treat the existing scar tissue. So, and the thing also we have bioprinted a human-sized corneal stroma, and we have characterized it. And now we are doing the in vivo studies to further understand the potential of this bioprinted cornea for treatment of blinding scar tissue formation, blinding scar tissue to prevent blinding scar tissue, also to treat the scarred blinding patient getting blind due to scar tissue formation. So the future scope, we are also working on different other things like that. We are also finding out the toxicology studies of this hydrogel as per the FDA and CDCRI guidelines. And also we are doing feasibility study on human corneal scar patient, that is we are going to start. So and then these are the things. So human hydrogel for ectasia, preclinical evaluation will be done on this thing, and a feasibility study on human keratoconus patient also. So these are the things are on the radar. So we are working on these things. These are the few references. Please look these references for further understanding, for further studying. I have used these references for this thing. Yeah. In acknowledgement, this work was supported. First, we have got a DBT grant to initiate this work, where we started the collaboration. So this is as I mentioned, this is a collaboration between LB Prasad Institute, CCMB, and IIT Hyderabad, where the scientists and clinicians from these three institutes are working together to develop India's first, and we have already developed India's first 3D bioprinted cornea. And so with the DBT support, we have started this project. After that, we were successful to get funding from ICMF for in vivo studies of our hydrogel, and also recently we got funding from Sri Padmavati Foundation, that they are supporting our clinical translation of this hydrogel, where we are targeting uh, to use this hydrogel for treating human patients with corneal scar and keratoconus. And I should also acknowledge my student, Dr. Shibu Chamitachal, who has done this wonderful work in collaboration with others. So thank you all for listening to this lecture. I'll be happy to answer any questions if you have. Thank you very much.